would like to.
Committee will come to order. I'd like to begin this hearing by stating the Oversight Committee mission statement. We exist to secure two fundamental principles. First, Americans have the right to know that the money Washington takes from them is well spent. And second, Americans deserve an efficient, effective government that works for them. Our duty on the Oversight and Government Reform Committee is to protect these rights. Our solemn responsibility is to hold government accountable to taxpayers because taxpayers have the right to know what they get from their government. We will work tirelessly in partnership with citizen watchdogs to deliver the facts to the American people and bring genuine reform to the federal bureaucracy. This is the mission of the Oversight and Government Reform Committee. I'm going to waive our opening statements today from the ranking member and myself because we have votes that are coming very soon, and I want to make sure that we get the opening statements from our guests that are here on the first panel. It looks like those votes will be called fairly shortly. When they are called, we'll slip away, vote, and then we'll come back and we'll do questions from then, and then obviously move on to our second panel. So glad to hear. But with that, I'd like to recognize Mrs. Spears. She has one of the panelists is from her district. Actually, I want to get a chance for her to be able to recognize them.
Mr. Chairman, thank you, and I too will submit my opening statement for the record. I do want to take great pleasure in introducing someone who I've known professionally for a number of years. He is the CEO of Oncomed Pharmaceuticals, and that's Paul Hastings, who is with us this afternoon. Oncomed is located in my district and is doing groundbreaking work on stem cell therapies that could provide important alternatives for the treatment of cancer. And Mr. Hastings is what you would refer to as a serial startup CEO and has done great work over many decades. So thank you, Mr. Chairman.
All members will actually have 7 days to be able to submit opening statements for the record as well. Let me introduce the other two panelists as well. We have two prolific writers that are here. Dr. Scott Gottlieb is the resident fellow at the American Enterprise Institute, and Mr. Peter Huber is the senior fellow at the Manhattan Institute. All done significant work and research in the area. If you're not familiar with our topic today, we are dealing with the FDA and the drug approval process, and so glad to have all three of you here as experts in this conversation. So pursuant to committee rules, all witnesses are sworn in before they testify. So if you please stand, raise your right hand. You solemnly swear or affirm the testimony you're about to give will be the truth, the whole truth, and nothing but the truth, so help you God. Thank you. You may be seated. Let the record reflect all the witnesses answered in the affirmative. In order to allow time for discussion, I would ask you to keep your testimony, your oral testimony, to about 5 minutes. You'll see the clock in front of you. All of you are veterans at this table before, and so we'd ask you to do that oral statement, as you know full well. All of you have submitted a tremendous amount of written information as well. That will go into the permanent record also. Mr. Gottlieb, be glad to be able to receive your opening statement.
Thanks a lot, Mr. Chair, Mrs. Ranking Member. Thank you for the opportunity to testify today before the committee. My name is Scott Gottlieb. I'm a physician and resident fellow at the American Enterprise Institute. I previously worked at FDA as the agency's deputy commissioner and at CMS as a senior advisor to the administrator. I want to address today the issues related to FDA's review and approval of novel treatments for serious diseases that aren't adequately addressed by available medicine. The FDA has been effectively implementing provisions included in the last reauthorization of the Prescription Drug User Fee Act related to breakthrough therapies. I believe these provisions are having a noticeable impact on FDA's willingness to embrace new approaches to expedite the development of these sorts of new treatments. But I still believe there's more that can be done. The drug development process itself has become long and costly owing to regulation that serves to add to pre-market burdens, but often without meaningfully improving the safety of drugs or what we know about their effectiveness. And we're not taking full advantage of what science has made available, not only in terms of new and more targeted therapies, but better ways for evaluating them. The review staff at FDA is a dedicated and well-intentioned clinical group of people who are often leading experts in their respective fields, but they are also heavily influenced by outside voices, and it's often the critics talking the loudest. Years of complaints about FDA's oversight of drug safety, about the high cost of drugs relative to their perceived benefits at the time of initial market entry, and criticism about the science that FDA uses to review to its review processes from vocal academics who often have their own parochial views in these matters. All of these things have taken a toll on FDA's culture. Over time, it's standing down people's willingness to take the risk of adopting new approaches to the agency's work, even around areas of unmet medical needs that might not have anything to do with the concerns that incited the initial concerns. The result is that a fear of uncertainty now pervades the review process. When it comes to drugs targeted to unmet medical needs, I believe it's a fear of uncertainty around efficacy that's having the most profound impact on how drugs are being developed. FDA staff is often unwilling to take risk when it comes to observations around drug efficacy. They require experiments that leave little doubt that the magnitude of the benefit being observed in a trial is not a function of any statistical chance. In short, they want to conduct pristine experiments that leave little uncertainty about the results describing a drug's efficacy, that they're precise and beyond any statistical doubt. Here it's important to distinguish between the magnitude of the benefit being observed and the believability of that result. I'm not talking here about FDA's concern that a drug must show a certain amount of benefit. Some threshold of measurable benefit is always necessary to provide a proper balance against known risks. Rather, it's how FDA guarantees the believability of that observation of benefit that I believe is having the most significant delay on the development of new drugs. FDA requires longer, larger trials to get pristine statistical results. This makes their ultimate decision around the approval easier since evidence is clear, but it also adds to time and cost. I believe there are ways to enable faster development of new drugs for unmet diseases and timelier access while still ensuring that future patients will have appropriate information. We need to focus on reforms that will help the review culture at FDA evolve when it comes to these issues, and I want to offer some suggestions that are aimed towards these ends. First, we should consider changing how clinical effectiveness is defined in the setting of rare diseases. FDA insists that there is a single standard for establishing safety and effectiveness. I think the FDA needs clearer direction around when we as a society wanted to exercise its existing discretion to streamline development programs. This doesn't mean that safety and effectiveness isn't firmly established for the drugs aimed at rare disorders. It only means that we are making a much more explicit acknowledgement of FDA's existing discretion to adjust trial requirements based on the circumstances. Second, the Breakthrough Therapies pathway has been a successful legislative effort, and its implementation by FDA has had a palpable impact on the review process. But a full-throated embrace of the spirit of this legislation requires a cultural change at FDA that is invariably slow to unfold. For these reasons, we might also consider changing the organizational structure of FDA to hasten the adoption of these provisions. Specifically, rather than allow drugs aimed at very rare disorders to be reviewed alongside drugs targeted to more common maladies, we might consider carving out the novel breakthrough drugs into a separate group inside FDA, a sort of skunk works charged with implementing novel review requirements in regulatory science. Such a group might more readily embrace concepts that can change how we develop drugs, introducing greater efficiency, ideas like the use of adaptive trial designs, Bayesian statistical techniques, and wider use of molecular profiling and targeting medicines. These are not new concepts, but they are very slow to gain adoption. In an agency where reviewers are under constant political pressure and time constraints, they don't feel a lot of liberty to incorporate unfamiliar and new approaches or take new risks in embracing concepts that are untried. So they stick with familiar constructs, even if these traditional approaches are unnecessarily costly and burdensome. These are just a few ideas on how to advance FDA science and make the process for the consideration of drugs aimed at vexing and unmet medical diseases more efficient. FDA has made great strides towards these ends through its recent implementation of breakthrough therapies. I would argue that to make further reforms, it would require measures that start to change the culture of FDA as it relates to these challenges. Thank you.
Mr. Huber.
Mr. Chairman, Madam Ranking Member, among federal regulators, the FDA plays a uniquely strong role in regulating not just the product, but the development of the core science that allows the industry to design the products that do what we want them to do. The science at stake here is not drug science. There is no such thing. It is drug-patient science. It is how the drug chemistry interacts with the patients. And drug designers can learn quite a bit by just studying biology, but at the end of the day, to get the science right, you do have to start prescribing the drug to patients and study what happens. So before it licenses a drug, the FDA issues something called an investigational license that scripts how we set about systematically and scientifically developing drug-patient science. And those scripts have simply not kept pace, in recent years particularly, with what the best scientific investigations can now do and should be doing. The blinded, randomized trial protocols that the FDA still relies on overwhelmingly to this day were first used in 1938. They were expanded and formalized in the 1960s. They begin with conventional clinical definitions of the disease. The criteria used to select the patients to participate in the trials must be specified before the trial begins, or to a limited extent resolved in the very very early phases of the trial when very few patients are involved. The doctors involved aren't allowed to systematically explore molecular factors that affect how one patient may respond well to a drug and another may respond badly to the same drug, even if they're presenting the same clinical symptoms. The only issues that these protocols address systematically are something called selection bias, which is deliberate or inadvertent stacking the data by doctors, or the placebo effect, which is wishful thinking by patients. The trials teach us next to nothing about how variations in patient chemistry affect responses to the drug. The FDA's concerns about selection bias are legitimate, but modern molecular medicine hinges on the deliberate scientific selection of the right drug-patient molecular combinations. Patients suffering from the same clinically defined disease often present different clusters of molecular targets deep down. There is no such disease as breast cancer. There are at least 10 biochemically distinct breast cancers down there. We treat some with estrogen blockers, and we treat others with estrogen itself. One of the estrogen blockers, its performance depends on liver genes, which determine whether the patient metabolizes that drug properly or not. And I could go on and on. There are diseases that change rapidly on the fly. The chemistry of cancer cells changes at a wild pace as it progresses. So do HIV infections. Many of these fast-changing diseases require multi-drug regimens and cocktails. Side effects add still more relevant patient-side variation in the chemistry. A good trial of a good drug should culminate in prescription protocols that will make it possible for future doctors to prescribe the drug to the patients who present the molecular profiles that will interact well with that drug. When we don't do that, there are two major consequences. The first one is little noted but deserves a lot more attention. During the trials themselves, we may quite often be doing real harm unnecessarily to significant numbers of the patients involved. A consensus report issued a few years ago by a coalition of cancer experts drawn from the industry, academia, and the FDA itself, says the FDA still relies on, and I quote, traditional population-based models of clinical trials, ellipses, that may form the antithesis of personalized medicine, and accordingly, these trials expose large numbers of patients to drugs from which they may not benefit. If you are saying that you are exposing large numbers of patients to cancer drugs from which they won't benefit, you are saying quite something, because these drugs are very often toxic and powerful drugs, and you really don't want to be prescribing them to the wrong patients for long. The second consequence follows directly from the first. If we are testing drugs in many of the wrong patients, many drugs that we do in fact need because they would benefit significant numbers of patients will not make it through these trial protocols, because to perform well, a drug has to be prescribed well. We know how to design what are called adaptive trials. In brief, you gather a great deal of data tracking genomes and proteins and other biomarkers that may affect the trajectory of the disease and side effects and cause different patients to respond in different ways. Patients can be added or removed from trials, or treatment regimens can be altered to improve our understanding of the drug-patient molecular science. There are sophisticated statistical profiles, processes and algorithms that can handle this very complex data. Because the adapters learn, and because the investigators learn and adapt as they go, the patients involved receive, on average, much better treatments. It really is high time to dispense with these old trial protocols and use the statistical methods of the future and the modern molecular tools that let us track and learn about all these factors.
Thank you. Mr. Hastings. Mr. Hastings, do you mind turning your microphone on there? Thank you.
Sorry about that. Chairman Langford and Ranking Member Spear, members of the committee, my name is Paul Hastings, Chairman and CEO of Oncomed Pharmaceuticals, headquartered in Redwood City, California. I also serve as chairman of the Bio-Emerging Companies Section Governing Board, which represents the small entrepreneurial and emerging biotechnology companies that often do not yet have a product on the market, and we are the majority of BIO's over 1,000 members. I personally have 27 years of experience in biotechnology and the pharmaceutical industry. My current company, Oncomed Pharmaceuticals, is working at the cutting edge of oncology research focused on antibodies that target a specific set of cells within tumors known as tumor-initiating cells. These cells drive the growth and metastasis of the tumor, the spread, and they can differentiate into various cell types within the tumor. Currently, we have 5 products in clinical development, all discovered at Oncomed, in over 13 completed or ongoing clinical trials, with more than 280 patients receiving our investigational agents. We continue to pursue the discovery of additional disruptive and novel anti-tumor initiating product candidates. The U.S. biotechnology industry is working on treatments and therapies that have the potential to deliver new solutions to our most pressing healthcare needs and is a key element of an innovation-driven economy. We've come a long way in turning incurable disease to treatable disease, increasing the ability of patients to maintain independent lives. With the number of people over 65 increasing, improving the quality of life and ability for patients to maintain independence is a national imperative. Hundreds of companies like mine are working on these solutions, with over 400 clinical trials currently underway focused on developing the next generation of medicines for over 200 diseases. This is also an industry poised to be a major contributor to a 21st century innovation-driven economy in the United States. However, we continue to face intense competition from other countries, as well as increasing R&D costs, regulatory challenges, and a contracted funding environment. This year, working with the FDA, we've seen positive signs that the biotechnology industry is recovering, not only from some of the regulatory hurdles, but also from the economic crisis. 39 biotech companies have gone public this year, including my own, marking the most active IPO market in a decade. Additionally, in 2012, the FDA approved 39 new molecular entities, the most approvals we have seen in 16 years. Now, while this is good news, the financial and regulatory environment continues to pose significant challenges to innovation and drug and biologic developers like ourselves. For example, first-time private financings, not public financings, but private financing, venture capital financings, the lifeblood of the innovation biotechnology industry. So these first-time VC financings for new companies are actually at the lowest we've seen since 1995. And while we've seen an increase in the number of approvals, we've also seen a steep increase in research and development costs, much of which is associated with increased requirements and costs to run clinical trials. FDASIA contains several provisions designed in cooperation with industry and the FDA specifically to provide FDA with the resources and processes that encourage the utilization of modern tools and approaches, such as adaptive clinical trial design, and allow for more interactive scientific dialogue between the FDA, the industry, and patients. Some of the most exciting provisions in FDASIA now include an expanded accelerated approval pathway designed to improve on the historical success this program has had with developing game-changing medicines to treat HIV and AIDS and other cancers, as well as other diseases, and expand its utilization in other disease areas, and a new breakthrough therapy designation designed to get the most promising medicines to patients much more efficiently. However, while FDASIA included an agreement by the industry to significantly increase user fee funding for the FDA to help support FDA's drug review activities and enable them, and these new programs, sequestration has diverted a portion of these industry fees to an escrow account that has no practical purpose for the FDA, industry, or patients. This has severely hindered FDA's ability to fully implement critical provisions of FDASIA that would improve our ability to more effectively develop and deliver innovative medicines to patients. This is the equivalent of our paying our utility bills and then being told we can't access power, lights, or heat. BIO would like to thank Congress for proposing a 2-year delay in sequestration of user fees in the proposed budget agreement, but urges Congress to rectify this irrational and counterintuitive situation by passing the FDA Safety Over Sequestration Act of 2013, sponsored by Representatives Leonard Lance and Anna Eshoo, with broad bipartisan support. While many new measures have been embraced and encouraged by Dr. Hamburg, Dr. Woodcock, and their colleagues in management at the FDA, it's yet to be seen if the regulatory flexibility afforded by FDASIA is being fully embraced at and across the FDA reviewer level to advance the development of new therapies for unmet medical needs. Releasing user fees from sequestration, successful implementation of FDASIA, and enabling our colleagues at the FDA could significantly improve the ability of our industry to more effectively develop new medicines and get them to patients who need them. These actions could also help stimulate investment in early-stage companies that are working on the next generation of medical discoveries and breakthroughs. The Biotechnology Industry Organization is committed to working with FDA and Congress to ensure these goals are achieved. Thank you for this opportunity, and I look forward to your questions.
Thank you. I'd recognize myself for the first round of questions, and we will go through the questions as we can. And we're still not called for votes yet. You'll hear the bells and the lights and everything else go off when that happens, and we'll have a little bit of time to be able to move from there. Let me ask a couple quick questions on this. I think I'm going to work in reverse. Mr. Hastings, you talk about the venture capital being the lowest it's been since 1995 on that. Do you have a gut feeling on why that is? Why is the venture capital suddenly drying up when we have a record number of IPOs happening?
It's a cycle. So while now the public markets are doing well, the venture capitalists who have fueled all these early-stage companies had fueled them for so many years before the IPO market was open that they drained themselves of a lot of their resources. They weren't able to provide the returns to their investors. So when the public markets opened up and the public investors embraced the public markets, everybody on the public side did really well, but there's a lot of catch-up that's being done now by the venture capitalists. So they now look like they haven't provided their returns over the course of the 10 or so years prior to the public market opening up that their investors required. So those same investors invest in the public markets as well.
Okay. So what's your gut on how quickly that turns around?
I think it's going to...
You've tracked this for a while, it sounds like.
Yes, I track this constantly. It's going to take a few years. So once the public markets continue to reward the venture capitalists for delivering these companies. So now, by the way, all those private investors, all the investors that are in Oncomed Pharmaceuticals on the private side, they won't be able to cash out until the lockup period is over, and even then, they have to see the company do well on the public markets in order for them to get their exit. All that takes a little bit of time, probably a couple years before people start reinvesting now in the early stage venture financing.
Mr. Huber and Dr. Gottlieb, they're both recommending different ways of doing some of the trials and processes and things, and I want to be able to get to both of those in a moment. Mr. Hastings, though, what is the most expensive part or the most difficult or cumbersome part of the clinical trial or the R&D or approval process? You can broaden that as broad as you need to make it, of getting the actual drug through the research to development. What is the pricey, cumbersome part of that?
I'll give you one example. There are many places along the development cascade where it gets expensive. In the early stages, for young companies, it gets expensive even in the phase 1 or phase 2 portion of the trial, particularly in phase 1, when there's a lot of communication back and forth with regard to safety of the drug. And one of the areas that we've been working on is this informal communication, where rather than having a letter writing campaign back and forth, there's more dialogue between the reviewer and the company. Saving a month on that communication process could save a small company. And there's been a lot of effort from both the FDA and the industry to work on that together. That's been an example of something that's improved dramatically.
Okay. And Mr. Huber, let me ask you a quick question on it as well. This statement is interesting to me, the molecular profiles. How would that work for them, and expand that somewhat is what I'm saying, as far as the FDA, because that's obviously a completely different paradigm than what they're currently doing on it. So how would that function in real life for them?
I was having dinner last night with a prominent oncologist from MD Anderson in Houston, and he said the real clinical trials begin after the drug is licensed. If you look at how oncologists, now I will emphasize first of all that cancer is a very distinctive disease. It's wildly dynamic. The cells are constantly mutating, and there isn't, by the time a tumor grows at all, you don't have one disease in there. You've got a whole bunch. That's what makes it so hard to attack it, and why so often drug cocktails are used and so on. But in any event, the fact is that oncology has been the beneficiary of the accelerated approval quite often. It's one of two diseases. The other are HIV infections have gotten this. And the main advantage of accelerated approval in my book is that it releases oncologists to practice and to work out what the patient needs step by step. The answer is you begin prescribing drugs. These are targeted drugs increasingly that target one molecular receptor. You can track very early on with modern diagnostic tools what's going on, not up at the clinical level, which takes quite a lot longer to to surface, but much deeper down inside the patient's body. I mean, it can be things that are not that much deeper down, like tumor shrinkage or so on, but you can also be looking at densities of cancer cells being shed from the tumor and circulating in the patient's blood. You can get lots of the FDA calls these surrogate endpoints or intermediate endpoints, and you get feedback much faster. If you begin getting that, you can early on begin saying, look, we begin seeing different patterns of response, and it gets eye-glazing when you talk about the statistics, but the fact is the statisticians know how to handle multidimensional trials where you're exploring multiple factors as you go. We have mechanistic understandings of why certain receptors should be accelerating the progress of the disease, or if you inhibit them, slowing it down. You study these things and you adapt. I could give you one vivid example. If you'll allow me to. Memorial Sloan Kettering in New York some years ago launched a trial using a kidney cancer drug to treat bladder cancer. It was an abysmal failure. Almost everybody who was treated with this drug did not respond well. However, one 73-year-old woman responded extraordinarily well. In fact, 2 years later, she's completely cancer-free. Now, in the past, and certainly if this were all within the confines of one trial, you'd ignore that. I mean, one patient does not license a drug. The Memorial Sloan Kettering doctors went in and they searched for the biomarkers associated with cancer that explained this. They couldn't find one. They then did a whole tumor sequence. They just looked for everything that was in this one patient's tumor, and they found the receptor that was being targeted by the kidney cancer drug. One patient in that group. It turns out about 8% of bladder cancers have this receptor, and so then they launched a new trial. That is a slow-motion adaptive trial. You learn something and then you change. You can do that internal to a single trial if you're working hard at it. But you have to do things that the FDA doesn't allow. You actually have to be saying, these are the people who are being treated with a drug. Let's look at what's the details. I mean, if you have toxicity problems and metabolism problems, same thing. We've got biomarkers for livers that metabolize things well or badly. You can look for them, and you can then converge on treatment protocols that work better.
Great. We'll follow up with FDA in the days ahead. Let me yield to Mrs. Spear for her questions. They have called votes now. We have around 11 minutes or so on the vote count. I think what we'll do is take Mrs. Spear's questions, then we'll take a quick recess. We'll have 3 different votes that are happening, and then we'll come back and we'll go at you with some more questions. You okay with that? So you'll get a quick break. Members on the dais, Mrs. Spear.
Mr. Chairman, thank you, and thank you all for your testimony. The sequestration has really cut the limbs off of NIH funding to the tune of about $1.2 billion a year. From your perspectives, each of you, I want to know how that affects your ability to do your science.
Well, that's a wide open question. So to me, it goes all the way back to what and how we want to be perceived as a nation, right? If we're not getting behind STEM education from kindergarten through grade 12 all the way up through our university system and the NIH, and getting behind innovation, it's not then going to leave the technology transfer offices of our universities or the halls of the NIH and come with a license to an entrepreneur who then can develop that drug. So it's a little hard to pinpoint exactly what sequestration does with the NIH in terms of what it might bring to our industry, other than to say it's huge very broadly. You can't really look at any one drug that came out recently and say that wouldn't have happened under sequestration. I can't think of one off the top of my head, but that's the kind of thing that comes out, the thought process, the focus on innovation, and without that, and without those jobs for those people to do those things, they're going to go to other places.
I'm less focused on NIH, just to pick up on what Mr. Hastings said about FDA. I think the sequestration has been problematic for the FDA insofar as what I see, and I wrote an article about this. A lot of the sequestered funds were funds that would have been targeted towards the areas of new regulatory policy making, the things that were embedded in PDUFA in particular, where FDA was going to advance or try to advance some of the scientific principles that we're talking about here today. There were unfilled positions at the time that the sequester was imposed that will continue probably to go unfilled, but a large chunk of the money that was taken out of the programs of new regulatory policy making.
Dr. Gottlieb, you suggest that there is a certain amount of risk that we should just be willing to accept. And I'd like to know what you think that percentage of risk should be in terms of the FDA process.
Yeah, I'm not sure you can, articulate it in terms of a percentage. There's obviously a certain level of palpable risk we're always willing to tolerate, and it adjusts based on what the clinical circumstances are. And if you're facing a grave disorder and you don't have other options, you're willing to embrace quite a bit of risk. I cited in my written testimony the case of mucopolysaccharide diseases that are inborn errors of metabolism where children are born. These are largely fatal diseases. They're terribly debilitating. I think families who have children with those disorders would be willing to embrace a large degree of uncertainty around a new drug, but in many circumstances today they're not able to because the clinical trial requirements are getting more difficult. Remember, I'm talking here about, in particular, risk around the benefit, not the safety. I'm talking about situations where the magnitude of the benefit that's observed in a clinical trial can't be firmly established because the statistical rigor hasn't reached a high enough degree of sureness, if you will. So you see a certain magnitude of benefit, and you have to probability adjust that and say, well, since it wasn't a large trial and there were these flaws in the trial, we can be 70% certain that it's going to deliver that 40% benefit. So that's the situation.
But my understanding is that, and I've actually accessed compassionate use through the FDA for a number of constituents from time to time. My understanding has been that they have been very willing to allow the drugs to be used for compassionate use, which typically you would say in the case of a young child would be embraced. So do you find that at all being restricted in its availability?
No, I think FDA has been very flexible when it comes to individual patient INDs, but keep in mind there's two challenges there. One is that only certain physicians are going to be able to navigate that process in collaboration with their patients. It takes a certain level of sophistication and resources to work through that process, and that's not a criticism of FDA. It's just a hard process. So it is putting at a disadvantage a whole lot of patients who will never have access to that. And the other thing is that if that's what we're dependent upon to make therapeutics available in these kinds of sort of grave situations or situations where there's a real unmet need, the framework, the sort of framework on the industry side won't be in place to make the drug broadly available. In today's environment, especially with a lot of biopharmaceuticals, the manufacturing isn't available until the time of approval.
All right,
and so we're dependent upon that approval.
I'm going to try and get one more question to Mr. Hastings. Mr. Hastings, if there's one thing that you would recommend that we do to assist the emerging bio companies to be successful in their interactions with the FDA, what would that be?
Enable the FDA through some of the policies that we've put forth, some of the breakthrough therapeutic areas which would allow us to, with appropriate risk, also see the reward for these patients. So taking some of the risks that Dr. Gottlieb was just talking about. There are certain diseases like cancer. There are other diseases, chronic inflammatory diseases where patients recognize some of the risks associated with therapies. And so enabling some of these breakthrough areas where we can get drugs approved quickly, safely to patients faster is the way to go. And there's a number of those initiatives that I outlined in my testimony.
Thank you. My time is up.
Actually, all of our time is up for this point. I'd like to take a recess for about 20 minutes. We have 3 votes in this series. Each one of the votes is about 5 minutes apiece in between. We'll go over and do the votes. We'll come right back, and then we'll jump right back into pummeling you with some more questions, if that's all right with you. So let's take a short recess.